Hi everyone, welcome to my channel Pure Biology, and today I come with another video, that is the transcription factor. So this is also responsible for the eukaryotic transcription, and another two important thing, that is the RNA polymerase, and another is that promoter. Okay, so already I have explained in my previous video, so you just check out that one. And this is here I totally try to describe about the role of the transcription factors. Okay, so transcription factors is that this is the eukaryotes require many protein factors. Transcription of eukaryotes that require many protein factors, and these protein factors are known as the transcription factors. And these transcription factors, maybe this is made up of the protein, but it is different from the RNA polymerase. Okay, and to initiate as well, because RNA polymerase only trying to initiate initiate the transcription process, but here the transcription factor, this has two roles. One, it maybe initiate, and also it can regulate the transcription process. Okay, the T factors is the transcription factor. This is trans-regulatory and soluble protein and produced by the gene distant from the chromosome region that it affects. So what is saying that it is produced from the gene of the chromosome region. Okay, and this which gene is responsible for producing these T factors? That chromosome region have no effects on the transcription process. That means the distant the position where the transcription process is continuing, from there the distance place of the gene from where the transcription factors are produced. Clear? And these transcription factors, that is made up of the soluble proteins. Okay, that is made up of the soluble protein, and it attaches to the DNA at a gene regulatory site, and by doing so influences the rate of transcription of a specific gene. So in that way T factors or the transcription factor regulates me, that means it create an influence on the gene now to create the initiation process of the transcription, or start to initiation process. Clear? And here another important thing is that this attached with the DNA. Okay, DNA where? At the regulatory site, that means the controlled site, or the site which can control the transcription process. This is the regulatory site. Now the T factors, these have the two types. Okay, generally basal, and the another is the regulatory. So basal means general, or you can say that initiation process is controlled by these factors, and regulatory transcription factor, this generally control this regulates the initiation process or the transcription process. Now the regulatory sequence like the enhancer and the silencer. So here the transcription, okay, one thing is that basal transcription factors, these are the TF2D, TF2A, and etc., and which attach to where? The promoter. Clear? So basal transcription factor, where they try to attach? They try to attach to the promoters. And the regulatory transcription factors, these try to bind to the regulatory sequences like the enhancer and the silencer. Okay, here the two terms enhancer and silencer, meaning is that enhancer means enhance the initiation or the transcription process, and the silencer means that try to stop. So if this enhancer and silencer, I will try to explain this in another topic. Clear? So regulatory transcription factor that binds with the silencer or the enhancer, and the basal transcription factors that bind with the promoters. Clear? Now basal transcription factors that determine the transcription start site, I have already told that these bind with the start site. Why? Because to initiate the process. And the regulatory transcription factors that be repressor or the transcriptional activators. Repressor means that they will try to repress the process or stop the process or enhance the process. Clear? So now the transcriptional activators like the SP1 bind to regulatory sequences and stimulate the transcription. So here the one name of the activator is that SP1. This is called the activator, and this bind with the regulatory sequence. So regulatory sequence here play an important role because this regulatory sequence is the binding site for the activators and also binding site of the transcription factors. Now this is the example SP1. This is the example that bind with the regulatory sequences and stimulate the transcription process. Clear? So here these are the list of some examples, or list you can say, that regulatory transcription factors and the basal transcription factors. So regulatory transcription factors CBF, C or EBP, CREB, SP1, etc. So CBF means CAAT binding factors, and that binds to the CAAT box. C or EBP, that means CAAT or enhancer binding protein, that binds to the CAAT box. And CREB means CAMP response element binding protein, that binds to CAMP response element. And SP1, this is the SV40 early and late promoter binding protein, that binds to the GC box. Clear? Now the basal transcription factor is the TF2D. TF2D is TAFs for TAFs, TAFS for, and TF2D here the TBP component. And another TF2A, TF2B, TF2F and TF2E and TF2H, and these are the functions. Functions means TF2D, that its function is that recognition of TA box, and TF2D recognition of the core promoter, that means it can recognize, and TF2A that stabilizes the TBP and TAF binding, TF2B that influences the selection of the this is the start point, okay, start point. TF2F recruitment of RNA molecule, TF2E that modulates the activation of the TF2H, and TF2H that helicates the activity responsible for the termination. So these are the list and functions, and also the binding site is explained here where they can bind. So this is the very brief topic that is the transcription factor, and how this transcription factor play important role in the transcription. Here I just tried to explain briefly. Okay, so another important thing also responsible for the transcription process, and I will try to explain in another video. So continue with my channel, and thank you.